We are Isa and Shimako from Hirogakuen High School, and today we will be giving a presentation about our ongoing research that we have been working on, which is a comparative analysis of the cytotoxic effects of caffeine and theobromine in HEK293a cells. Caffeine is an adenosine receptor antagonist and is able to prevent adenosine, which is an inhibitory neurotransmitter that causes sleepiness, from exerting its functions. The chemical equation of caffeine is C8H10N4O2, and the chemical equation for adenosine is C10H13N5O4. And due to the similar chemical structure, caffeine is able to block the receptors of adenosine, preventing its activation. As a result, caffeine is able to make people feel awake and energetic. While a considerable amount of caffeine consumption is not harmful to the body, an excessive amount can lead to numerous negative effects, such as restlessness, insomnia, headaches, fast heart rates, and anxiety. Moreover, it is easy to build a dependence on it, making consumers require a higher dose of caffeine in order to get the same results. Theobromine, which is a metabolite of caffeine, has one less methyl group compared to caffeine as it gets removed during the process of metabolism. Theobromine is known to be found in foods such as chocolate and tea plants, and is known to have a similar effect as caffeine. However, the one missing methyl group makes the affinity to the adenosine receptor slightly weaker compared to caffeine, making it exert a weaker effect of boosting energy levels. It is also known to be less addictive and less harmful to the human body. We conducted our experiment in order to compare the cytotoxic effects of caffeine and theobromine on HEK293a cells to see whether or not theobromine has a significantly weaker effect, making it safer than caffeine. For our experiment, HEK293a cells were cultured and were transferred to two 6-well plates with approximately 100,000 cells in each plate. Solutions of caffeine and theobromine with a concentration of 0.1 millimolar was made, and into the 6-well plates, the 0.1 millimolar solution and the control solution were added. The solution in each well got mixed with DMEM that contained DMSO to solubilize the substances. After incubating the medium at 37 degrees Celsius for 48 hours, trypsin was injected into the wells to detach the cells from the plates, followed by a small amount of DMEM in order to stop the reaction of trypsin. Trypin blue was injected into each well using a micropipette, and the number of dead and alive cells were counted using a counting chamber and a microscope. A small amount of the solution was applied to the counting chamber, and the number of cells observed within the microscope were multiplied to approximate the number of alive and dead cells in the whole solution. This procedure was repeated 3 times for each solution for accuracy.
This is a bar graph showing the number of live cells after 48 hours of exposure to the solutions. The control showed the most number of live cells with the average of 11.67 times 10 to the power of 4 cells. The cells that were exposed to 0.1 millimolar caffeine showed an average of 1 times 10 to the power of 4 cells in each well. The cells that were exposed to 0.1 millimolar theobromine had an average of 3.89 times 10 to the power of 4 live cells in each well. The results showed significant difference in the number of live cells between the control and the other two. It can also be observed that theobromine had more number of live cells than caffeine, although not as significant as the difference between the control. This bar graph shows the number of dead cells in the well after 48 hours of exposure to the solutions. The control showed a significantly low number of dead cells compared to other wells with an average of 0.33 times 10 to the power of 4 cells. In the well where 0.1 millimolar of caffeine was applied, there were an average of 1.89 times 10 to the power of 4 dead cells observed. The cells that were exposed to theobromine showed an average of 1.44 times 10 to the power of 4 dead cells. The cells that were exposed to caffeine showed the least viability. Although cells exposed with caffeine had the most number of dead cells observed, there are no significant differences in the number of dead cells between theobromine. From this experiment, it can be said that both caffeine and theobromine have significant negative effect on cells' viability. However, these results did not confirm a clear difference of the cytotoxic effects that caffeine and theobromine holds. Although there were more live cells in the well that was exposed to theobromine compared to caffeine, the data we obtained could not fully support our hypothesis. The number of dead cells recorded in the experiment also showed that caffeine and theobromine were toxic to cells, showing more number of dead cells compared to the control. However, the number of dead cells between the well exposed to caffeine and theobromine were not significantly different, failing to support our hypothesis that theobromine is less toxic than caffeine. A possible source of error could have been due to inaccuracy caused during the pipetting process. Because the cells are very delicate, minor errors during the pipetting process could have easily affected the final number of live cells in the well. Because only one trial has been conducted, the results cannot be confirmed yet. The results failed to show significant differences between caffeine and theobromine, thus making our experiment incomplete. We will continue our research to aim to confirm the differences between their effect to find a less toxic alternative to caffeine.
Thank you so much for listening!